Gentlemen, recording started. So there are examples that we're going to highlight on responsiveness that come from your reading and study from Chapter 11. And then in regard to cell division, we're going to look at the types of cell division, namely mitosis, that was presented in Chapter 8. And we can relate that to that concept of asexual reproduction. Then the second type of cell division was meiosis, which you're studying now, which corresponds with sexual reproduction. So I'm going to highlight these areas that students tend to struggle with in relation to responsiveness and information continuity as you prepare for your quiz. Again, like Professor Pomerada said, if you have questions, please chime in, and we'll respond to those. Note the diagrams and the descriptions that we're going to go over, some of these together here, and then refer back to earlier discussions on cell division from the course that might help you just comprehend what's going on with meiosis here. So let's get into it here. Now the first concept that you encounter this week deals with the idea of responsiveness with hormones. So as we begin our review of Chapter 11, I want to highlight this term and ask you the question, what does this word mean? Can you describe the word hormone for us? And essentially, what maybe some of you are coming up with is that this is a signal molecule, right? It's something that's used to facilitate some type of a response. And your text gives this example of the hormone leptin, and we observe the leptin binding to hormone receptors, places that receive this proteins lying in the brain. So I was trying to connect for you the idea of how hormones are actually conducting a signal with the example of shall we eat, shall we not eat, do we need food, do we need that? And these hormones, these leptin hormones, for example, help to control weight. So it would seem appropriate that they would originate then from fat tissues, places where your body stores fat, lipids, and energy storage centers that we see in Figure 11.1. So a question for you to consider as you're studying this part is, what would you expect in your appetite if leptin levels were increased in your bloodstream? And conversely, what would you expect in your appetite if leptin levels decreased in your bloodstream? So connect the idea of the concept of a hormone to your body's ability to respond. And the purpose here, again, is to convey some of these principles of life that this course is based on, this idea that life is responsive. There are multiple other responsiveness descriptions that you'll want to key in on in this module. For example, the concept of homeostasis, like I already mentioned, and ask yourself, can you define and describe this term? And essentially, this term means that we're maintaining both the physical, again, and chemical conditions in an organism. When that chemistry of an organism shifts, as it changes, there's then a tendency for an organism to return towards its original state. And there's a wonderful example of the Venus flytrap, one of my favorite organisms in the text. And we see the modification, the changes that take place of this wetland plant to obtain nitrogen, right? To obtain nitrogen, the element, by capturing and consuming other organisms. A few questions to consider as you're looking at this in this concept of responsiveness. First, what is the sequence of the events? How does it respond at the cellular level, right? Within the plant cells, what type of a response is taking place? And then what's the specific role of the enzymes, the proteins that help move this along, right? And that type of enzyme that's described here is that of expansion, and as the name kind of describes, this ability of the cells to expand, and by expanding, it forces the trap closed. Right? It forces it closed, for example, around a fly. And as you study this, your text also points you back to previous study that you've done on nerves and that nerve impulse, the signaling in a nerve. That's why I have the nerve cells here. But we remember in the axon, there was that rush of ions that moved across the membrane, right, to send on that electrical signal. And in the sequence here of a flytrap, this leaf closes around an insect when there's a trigger hair that's moved. That starts off this reaction, if you will, and these trip hairs initiate the rush of ions across a membrane. If we recall membranes, the phospholipid bilayer, right, the membrane, which then promotes an electrical signal. This movement of ions similar to what we see in a nerve cell. So the electrical signal results when these outer leaf cells then begin to swell, right? There's a swelling in relation to the expansiveness here that takes place. And as they swell, that outer epidermis swells, it forces the leaf closed. It just pops shut. So from your study of plant cells, you know that they have walls, right? Cell walls. And the cellulose, the carbohydrate cellulose of these cell walls are effectively loosened by the enzyme or protein called expansin. So, for example, the expansion of the cells along the outer epidermis is what it's referring to. So when this occurs, water is basically rushing into those cells, expanding them to close the trap. So again, another form of response. There's a stimulus, right, with the trigger hair, leading to response linked to this idea of expansion proteins. So be sure to review how this trapping mechanism works. If we continue this idea of responsiveness, so we just look at the cellular level, and we can zoom out a little bit more and look at the organ system level, right? Responsiveness in the organ system level. And this idea is a response to environmental change. And your text talks repeatedly at this part about how our nervous system, our central nervous system, and that's why I have the brain here, how it's responding to light and dark levels. This is figure, I believe it's 11:18 in your text. So when we consider these concepts in relation to our brain, our central nervous system, we can identify multiple regions that are pointed out in your text, right? Different parts of the brain that help to regulate these daily alterations that we encounter in the light-dark cycles. Excuse me. For example, we have what are called suprachiasmatic nuclei. This region inside the hypothalamus, suprachiasmatic nuclei, that are helping with our response to changes in light and dark. It's a region in the brain there. This response in the nervous system, right, the brain's a part of the nervous system, leads to a stimulation, the pineal gland, right, another region of the brain. The pineal gland then secretes what's called melatonin. So all of these things are connected. Again, the concept of responsiveness in this case at the organ system level. So melatonin is important here in our discussion. It's another hormone, like we saw with leptin. It's a different type of a signal molecule, and this one specifically regulates the response to light and dark. Now, there's multiple tables in your text that highlight some of the usefulness of melatonin, and this is a result of the nervous system, which then fluctuates the melatonin levels in our bloodstreams, responding to light and dark. And this controls also how much melatonin makes contact with the suprachiasmatic nuclei, and it regulates the secretion from the pineal gland itself. All these levels are being regulated in response to light and dark conditions. Also note in your text that it details the influence of a hypothalamus, that region of your brain, on additional hormone signals. For example, it talks a bit about the thyroid gland. So this is on page 363. So you might be thinking to yourself, what does all this mean? All right, this idea of responsiveness. So we have hormones that act as signals. The melatonin levels, for example, help to generate this concept of wakefulness, our ability to wake up, right? The wakefulness. The table on page 365 in your book also suggests that taking supplements of melatonin can help with things such as insomnia or even depression and anxiety. So you could ask the question, are hormones an important design feature that reveal how life is responsive. And we say, yeah, the hormones help us, and they're an excellent example of this concept of responsiveness. And another form of responsiveness that was given to you is that of the transcriptional level. We can start by just saying, what is transcription? In your study, from DNA, we go to RNA, and from RNA we can make protein. And this transition from DNA to RNA is what we call that transcription. Right? So we call it transcription is a process of making a strand of nucleotides, a monomer, nucleotides from DNA. Going from DNA nucleotide strands to RNA nucleotide strands. And specifically, transcription would be the formation of this strand of RNA that can then be translated from RNA into protein. DNA to RNA to protein. And we call that a type of proteins that are essential in the metabolism. That word metabolism meaning all the sum of chemical reactions going on inside of us and in our cells. Those important proteins are called enzymes. Very important for metabolism. In your text, the example of enzymes being produced is an excellent example of responsiveness. So DNA you might think of as like a preserve, right? A preserve for, not like a game preserve for animals, but a preserve for biological information. Right? It's all housed in DNA. And one such example is found in the DNA of E. coli bacteria. So E. coli is the example given to us in your text. And there's a region of that DNA in E. coli that was identified back in the 60s or so that is called the lac operon. And that word operon is basically an on-off switch for a set of genes in the DNA. Right? Basically an on-off switch. So a region of the DNA is called the lac operon, which specifically is used to construct enzymes, proteins recall, enzymes that break down lactose. Lactose, as the name implies, that OSE at the end implies to us and tells us that's a type of carbohydrate. Right, type of carbohydrate. And there's what we call a repressor gene that represses, right, that holds back what is being produced. Right, so a repressor gene in the DNA strand, right, is being produced so that it is ready when lactose is present. So in the presence of lactose, we want to form enzymes that can metabolize lactose. By metabolize, we mean break it down, catabolize, break it apart into smaller pieces. So the repressor, as the name implies, it blocks the genes needed to produce enzymes for lactose metabolism. And in the presence of lactose, we're going to bypass that to form the enzymes. Again, a responsiveness based on these conditions that shows us this connection at the transcriptional level. So let's look at this further, the formation of enzymes in the lac operon. All right, so this is just continuing downstream from that image you just saw. All right, so taking that double helix, that spiraling strand, and then going beyond the repressor gene with the presence of lactose to form enzymes. So when the repressor protein binds to lactose, it changes its shape. And that shape is very important. So the change of shape of that protein lets go of what we call an operator, right? Like the name implies, right? It operates. It operates what's going to happen next here. The site of binding for what we call the repressor protein. So think of it as kind of like a blockade. There's really no point in making the enzymes for breaking down lactose if lactose isn't there. And with little or no lactose, the gene for lactose transport or lactose breakdown is not going to be formed. It's not going to be transcribed and then translated. When functioning, as we see in this image, a bacterium can then absorb and break down lactose if it has those genes. Like listed here, the genes for lactase, meiosis, transoxidase. These enzymes are needed to break it down further. All right, so this is an example at the transcriptional level, getting down into DNA itself. All right, so I want to shift to the next topic here. So let's shift to a second main topic, the idea of information continuity. We're going to get into the idea of how cells divide. So the continuity from cell to cell is of great importance in the discussion of life. What we're trying to preserve or pass on as the cells are dividing is what we're answering here. How can we describe each of the stages that occurs in the division of a cell? One of our questions we want to ask. Having a handle on mitosis from Chapter 8 is going to really help you move forward with this concept of meiosis. So our end goal in meiosis is going to be one set of chromosomes. So keep that in mind as we move ahead in the process. So let's look at a comparison between asexual and sexual reproduction as we begin talking about Chapter 12. So there's a nice YouTube clip of the paramecium dividing found in the course content this week, and that emphasizes this type of division, this idea of asexual, the paramecium dysaria in that image down there, going from 1 to 2 cells. So for a single-celled organism, cell division is a lot like reproduction. And we see multiple examples of plants and animals, like starfish or the budding of a hydra, that go through asexual forms of reproduction. And asexual reproduction is defined as the generation of offspring without the production by meiosis of haploid sex cells. We'll talk about haploid repeatedly here. Haploid, one set of chromosomes, and this without fertilization. So mitosis is also how our body cells, that's what we call our somatic cells. A teacher friend of mine likes to say with mitosis that mitoses go through mitosis, referring to the idea of body cells. Right? Mitoses go through mitosis. So mitosis is part of growth and also part of repair. Right? Like if you have an injury, that's how your body repairs itself through mitosis. Now, by contrast, sexual reproduction, the emphasis in this module here, is defined as generating offspring from the union of gametes. Not somatic cells, but gamete cells. The gametes are sex cells, right? Sex cells from two parents. By way of meiosis, gamete formation, sex cell formation, and fertilization, so the joining of those cells. So, you might want to, think about, what's meant by this idea of gametes or sex cells from two parents. And we'll talk about that. Also, what are the advantages and disadvantages for sexual reproduction? So in a biological sense here, sexual reproduction is going to increase genetic variation. Right? You're going to get genetic information from the mother and the father. One disadvantage that your text points out is that there's also a 50% chance that you can lose perhaps like a rare beneficial mutation that you have. It wouldn't be passed on. So we can review how that 50% chance would be reasonable. So let's connect some of these concepts. All right, so let's connect the process of meiosis, right, sex cell formation, gamete cell formation, to our own life cycle, and highlight some important terms in addition to mitosis and meiosis. So here we have the summary of the human life cycle, right? In this cycle where meiosis is the formation of gametes, right, sex cells, which are haploid, one N representing one set of chromosomes, one N representing one set of chromosomes, which then come together in fertilization, from the mother and the father, right? Two sexes of the same species shown here through meiosis coming together at fertilization to form a zygote, which would be then 2N, or what we would call diploid. We're going from haploid back to the diploid state, which is two sets of chromosomes. The zygote then undergoes mitotic division. We have this connection between sex cell formation and body cell formation, meiosis and mitosis being connected here. As the individual grows and develops into the multicellular adult like us, right? So one aspect that might jump out at you here is that reproductive cells differ from normal body or somatic cells. They're not exactly the same. So recall that somatic cells undergo mitosis, body cells go through mitosis, and here N represents a complete set of chromosomes. We're made up of trillions of cells, right? And in those cells, our genetic information is found. Where is this found? It's found in the DNA. And that DNA is coiled up into what we call chromosomes. And our normal body cells are diploid, or 2N. So we could say that they have two sets of chromosomes. This is also called diploid, di for two, and ploid in reference to the chromosomes. In humans, and us, typically we have 23 pairs, or what we would say 46 chromosomes. What would happen, consider what would happen if we did not reduce from 2N diploid to N, or haploid, in the formation of sex cells. Right? You're probably thinking this, and you'd be correct to say that we would double the number of chromosomes in an individual at fertilization if we did not reduce that number in half. Meiosis then reduces what we call the ploidy, reduces the ploidy level so that it can be restored when those gametes, the egg and sperm, unite. And with gametes, or gamo, quite literally means marriage, and gametes means that they will unite. So there are multiple disadvantages to sexual reproduction mentioned in your text as a result of genetic variation in the formation of gametes. Combinations of really good genes are not necessarily passed on intact to offspring. We'll see how this can be traced out with some of these ideas of division. We'll also see the important moment when the egg is fertilized by the sperm, which occurs when the male and female pronuclei, right, the life of the conceptus, talking about like our discussion on birth control, right, when the male and female pronuclei fuse together into one nucleus, a new life. This should make us wonder when we see the earliest origin of the individual, part of the discussion in your text. When the pronuclei fuse together, we note that the new individual, or zygote, now possesses the paternal father's DNA. The conceptus, then, is now a separate individual. So when we look at meiosis, we see a series of two divisions, or PMAT, you know, think of it that way to keep the order straight. Prophase, metaphase, anaphase, telophase. This is taking place within the gonad of our end goal event, being one set of chromosomes, so that when the pronuclei fuse together, we return to that diploid state, the 2N state. In meiosis, we find homologous chromosomes that are initially paired, and one homologous chromosome from dad and one from mom. And these chromosomes contain genes that code for proteins, right, like enzymes, for example, proteins, which may produce a visible trait. Alleles are different versions of the same gene. Like an example here, I put LUL and LUL, the capital and lowercase letters. So think back to mitosis. Do we see homologous chromosomes pair with one another during mitosis? No, we didn't see it then. Remember that in meiosis, we end up with genetic variation and not a clone of the starting cell as we saw in the example of mitosis. It's not a clone in that there's a swap of genes that takes place here. And so, as we saw in the cell cycle with mitosis, we have the prelude to mitotic division, in which chromosomes are replicated. And we get another copy. The first division of meiosis will result in 2 haploid cells. This is what we call a reduction division. Right? Prophase 1, and you want to keep all of these different phases straight and know what is happening in these different phases. And we have 2 divisions taking place here with meiosis. 1 with mitosis, but 2 with meiosis. So prophase 1 has a unique feature in that the homologous chromosomes actually swap pieces of information. As another colleague described, this is kind of like shuffling a deck of cards, right? And this then increases the genetic combinations. This is also the stage in which the centromere, this connecting point, the centromere of each chromosome can first be seen to attach to the 2 what we call sister chromatids. So after this crossing over event in prophase 1, we immediately go into metaphase. Metaphase 1, which is basically the homologous pairs lined up on opposite sides of what we call the midline in the cell, shown in the second cell down from the top. And that genetic variation is furthered here in that homologous chromosomes are oriented independently. They are oriented independently of each other during metaphase 1. We proceed to anaphase 1, right? Reduction division here, anaphase 1, and this is a stage of meiosis in which the total number of chromosomes is then cut in half, moving it from a diploid 2N to a 1N haploid state, diploid to haploid during anaphase 1. This may seem odd in that we have a 2-copy type of a situation, but those copies are going to separate again into the resulting haploid cells at the end of meiosis 2. So basically at this point, the homologous pairs are now separate. And then telophase or telophase 1 is a transition to prophase 2. So let me ask you a question. At this point, do the chromosomes unwind? Do the chromosomes during telophase 1 unwind at this point? Well, the answer would be no, because it's going to divide again. There's no reason to unpack all right now. It's all been packed up. It's going to divide again, and the spindle degrades, and it breaks down, it degrades, and the nuclear envelope is going to then reform. And cytokinesis, the splitting here, the cytokinesis follows right after telophase 1, and then we have 2 1N cells. So in humans, this would mean 23 2-copy chromosomes, 1 chromosome from each pair. Right? That brings us to meiosis 2, or the second round of division in meiosis. We're going to go from 1 to 4 cells, right? We started with 1, now we have 2, and then we're going to go and we're going to cut them in half again, split them again in meiosis 2. So we're seeing a multiplication of the possible number of gametes, sex cells, right? Division is essentially, at this point, the same as mitosis moving forward. But prophase 2, there's no crossing over. That was just in meiosis in prophase 1, during meiosis 1. There's no crossing over during prophase 2. It follows the basic pattern that we observed in mitosis. In metaphase 2, chromosomes are going to line up. They're going to line up along that plane of division again. In anaphase 2, those chromosomes are going to move apart. And now we have the chromosomes in a 1-copy, what we would call a chromatid state. And then lastly, in telophase 2, it's going to have 1-copy chromosomes that are in newly formed cells that we call gametes. And then cytokinesis ends with the formation, following telophase, cytokinesis ends with the formation of 4 haploid This is in contrast to what we saw in mitosis, right? So we went from diploid to diploid. Here, we're resulting in 4 haploid cells or gamete sex cells. 4 could be 4 sperm, or it could be, one of 4 of these developing into the egg or the ovum. So we end up with haploid cells.
Hey, Kyle.
Yes.
We have a question.
Great.
And the question is, does metaphase 1 cause reduction division?
Does metaphase 1 cause reduction division? Now, I'm noting reduction division specifically with anaphase 1.
And I would agree with that.
Yeah, I think that we don't see the actual reduction division until we get to anaphase 1. Because there we're moving from the diploid to the haploid state.
Okay, great. The student just wanted confirmation that that was correct, so I think we're good.
Great question. Good questions coming. Good question. Okay. All right. Well, let me just move on. If you have a question, please ask. Okay. So let me just move away from meiosis for a moment. Listen, if you have questions, just a few comments related to the idea of human development. We'll have a few questions potentially about that. But in human development, you might be wondering, when did these sex cells actually even originate? And when did these come about? Well, they originate as what we call primary germ cells. All right, and so these cells then later migrate through the rectal area of a developing individual to what we call gonadal ridges. So a location where then egg and or sperm would then be produced in that organism. So this region later develops into either what would be the testes or the ovary. So that's the site of meiosis taking place, right? Forming gametes, forming sex cells, all right? And then lastly, this idea of in your text about reproduction being constrained. You have multiple people talked about here how Aristotle considered the quickening, the start of life. Peter Singer there with some very some dark thoughts on the self-oriented individual. But in relation to meiosis, there are in this idea of constraints on reproduction. We should think about this in our worldview, right? Our Christian theism. How do we define an individual? We pointed out that the fusing of these pronuclei from the egg and the sperm results in an individual that's unique, right? Some individual that has genetic information from both the mother and also from the father. So important to consider to connect these ideas of information continuity and the mitosis, meiosis. Why do I need to know this stuff? Go with in a broader concept of why is life important? Why should we value God's design in individuals? So those are major thoughts coming from this module, and I'd be happy to go back and readdress any of these that you have questions on. Professor Palmer Hayes and I could both try to field some questions here if you have them.
So students, if you want to type into the chat box any questions you want to ask, or even just content you'd like us to review over again, go ahead and do that now, and we would love to tackle them for you. I did have one question from earlier that I'll go ahead and ask, and that is a student wanted just kind of a concise review of the lac operon, meaning, if lactose is present, this happens. If it's not present, then this happens.
Right. That's a great question. And so basically if I could summarize, and Professor Palmer, maybe you could chime in too, maybe say it in a different way might help them. But with the lac operon, we're saying operon, we're just saying this on-off switch basically for a set of genes. So the lack in reference to the presence of lactose. So in the presence of lactose, we're going to then transcribe, right? You're going to transcribe the genes for these specific enzymes, lactase, permease. So I don't know if that's going to really answer your question, but we're talking about with the lac operon a location in the DNA of E. coli that was identified to be basically turned on and used when enzymes were needed to break down to metabolize lactose. Again, this idea of responsiveness. So what the author, Dr. Detweiler, was identifying here is here's a concept of how responsiveness occurs within this concept of this biological principle in the transcription and what's happening between the DNA to RNA to protein.
And I think of it I like to think of an analogy like an assembly line, which is, some parts of it fit nicely with this and other parts don't, but it's just how I kind of view it with this on-off switch. So if you imagine if you're at a factory that's going to produce a product and you have an assembly line of workers that are working, then you have to have some kind of raw material present for the assembly line to even actually function and help to produce that product. If there are no raw materials present at all, there's really no point in going to the assembly line. So it's all about trying to conserve energy and all about efficiency, and as Mr. Harris said, about responsiveness to the fluctuations that are happening in that environment. So that's kind of how I view the lac operon as an on-off switch, depending on if lactose is actually present in the cell or not. Okay, so question just typed in is, it's a fill in the blank for us. If lactose is present, the gene will be transcribed. If it is not, the gene will not be transcribed, and what? And I think what she's looking for is translated, though you didn't really talk about translation in this presentation.
Right. Yeah, we didn't get into the translation part. I don't think the text here gets into specifics on translation. Actually, the formation of lactase, permease, etc. there. Are they talking about the blockade itself? So if lactose is not there, with the repressor gene, is that the question?
I'm not sure. She says it's actually a very specific question on the study guide. I think she's meaning translated, that if we're not going through transcription, then we're also not going to be going through translation from that gene.
Right. Yeah, we can't move forward if we're not going through transcription. So transcription is the prelude to translation.
Right. Yeah. So that was it. Any other questions that students would like to type in or review parts of the presentation? Well, it looks like we don't have any more questions. And students, you can always email your instructor, or you can email myself or Mr. Harris if you have further questions. And I will be emailing out the recording of this later on this evening or tomorrow morning. So with that, we'll say thank you for joining us, and thank you, Mr. Harris, for leading us through this review.
Oh, you're welcome. Thank you all.
Yeah, so I'm going to stop the recording